Greetings to everybody. Good morning, good afternoon, good evening, wherever you are. It's December 1st. It's the holiday season for many of us, and we're reaching the end of the year, so we'll be celebrating. We'll be saying Happy New Year to each other very, very, very soon. It's hard to believe. This is our last webcast for the year, and stay tuned about 2022. Just to remind you, we are recording this. Thank you. I'm Millicent Pitts. I'm the Executive Director of Ocean Exchange. You see our major sponsors here on this first page. Next slide. We exist to help advance the adoption of innovative solutions for healthy oceans, resilient coastal systems. And because of our major sponsorship from Wallenius Wilhelmsen, we also have some focus on zero emissions and the decarbonization of ocean shipping. Before we started this webcast, we were talking about all the ships out at sea off of Los Angeles, and that's made shipping more visible than it has been before, and we thank Wallenius Wilhelmsen for their sponsorship. We just completed a very successful ocean exchange event in Fort Lauderdale. We awarded $245,000 cash purse to 5 different startups. 2 of them got $100,000. I'm going to show one of the trophies. And there were 3 collegiate winners. 1 got $25,000 and 2 got $10,000. I encourage you to go to oceanexchange.org and read about these winners. Dane Dunstan, who is our MC today, is our MC in our live event. It was fun and challenging to have a live event, but I'm glad we did it, and we're moving forward. Is Zdenka with us?
I am.
Good. Okay. Just want to make sure you're here. I want to turn it over to you, and I want to say thank you for this collaboration with MTS this year. It's been fabulous. It's been great getting to know you. Thanks so much.
Great, Millie, and right back at you. We have really, really enjoyed this partnership, and we look forward to continuing our partnership in the new year. So for those of you who don't know Marine Technology Society, we have about 2,000 members in 20 countries, and we were established to make real contributions in fostering progress in marine science for the benefit of mankind. And our mission is to facilitate a broader understanding of the relevance of marine technology to the wider global issues, promote and improve marine technology and related education programs, and advance the development and tools and procedures required to explore, study, and further the responsibility and sustainable use of the oceans. We connect through our committees, which are both professional committees and sections, including a student sections and our early career ocean professionals. We are, like Millie, starting to get back into our events. We had a very successful Oceans Conference. It was Oceans 2021, San Diego, Porto. So it was a worldwide, both in-person and hybrid. And we're looking forward to our Oceans in Action Port Security event in March, our buoy program in April, and our Oceans Chennai in February. So we hope that you can join us. Our events will stay hybrid to be able to connect with as most people as we can. We talk about that we're a catalyst. We have launched our Marine Science and Technology Certification Program, both CMAR Sci and CMAR Tech. We encourage you to apply. Please go to our website to find out how you can do that. We also have a Women in Leadership in Science and Technology program where we have hosted a number of successful panels, and we have a webinar series. We communicate on social media, but we also communicate through our journal. And the June journal was all about the Ocean Decade and Ocean Shots, and that one is available to you at no cost. So please visit us at Marine Technology Society. And one final point, we are advertising, have advertised, and we are looking for a new executive director. So if you have a background in the marine tech, ocean industry, academia, and you want to work for a great organization, please apply. So with that, Dane, I'm going to turn it over to you for a fabulous program once again. Thank you.
Thank you. Looking forward to it. We've got a great one today. I'm very excited about this. When I was a little kid, probably about 9 years old, my mother was reading Anne Morrow Lindbergh's Gift from the Sea, which is a wonderful book. And it happened to coincide with us taking a trip down to Florida to see an aunt who lived there. And afterwards, they surprised us by taking us over to Captiva Island. And we spent some time there. That's where she wrote the book. I always associated that with seashells because that's sort of what the book was written about, but in a deeper sense, what the ocean gives us in terms of how we think about it when we're there. And she talked a lot about love and peacefulness and all of that. When we talk about the gifts of the sea, we think of, like I said, seashells, we think of seafood, and we think of fish. But what we're learning now is that there's so much more under the surface. There's so much more to discover and learn. We've spent a lot of time looking at this in the past. It's been very, very exciting to look at what's coming out of it. We've got two really, really great academics with us today who are doing enormous work in looking at how to get really great medicinal services out of there, and going to do great stuff. So let me start by introducing Dr. Shirley Piccone from the Harvard Branch Oceanographic Institute at Florida Atlantic University. Why don't you set the stage, Shirley, about what's going on and what we can learn?
Thanks, Dane. Here's the first slide, please. And actually, we can go to the next slide. I'm going to talk today about the biotechnological potential of deep sea sponges in particular. And I want to just start out by saying that there are, over the last several years, there have been a number of policy documents that have been written about the blue economy and the potential, the promise of marine biotechnology and drugs from the sea. And many of those actually now have been realized. And it's been estimated, in fact, this is an old number from 2019, that the annual value of marine-derived pharmaceuticals is more than 2.3 billion US dollars. Next, please. But I'm going to focus my presentation today on exploration and discovery of sponge-derived drug leads and bioproducts, as well as the challenges with the approaches that we're continuing to use for biodiscovery and development. So why sponges? Well, I've been working on sponges for my entire career. There are more than 9,000 described species. It's estimated that there may be as many as 25,000, probably even more. We're discovering more new species each year. Sponges are incredibly diverse. That biological diversity really translates to chemical diversity, and that chemical diversity is what underpins the potential for discovery of novel chemicals that can be developed into drugs as well as bioproducts. Next slide, please. So the way we and many others have been discovering drugs from the sea, exploring marine biodiversity for drug leads, is by going out to sea, collecting the sample. In our case, we were very lucky to be able to have access to the Johnson Sealing Plant submersibles, which allowed us to go as deep as 3,000 feet and to very carefully collect all or part of an organism without damage to the habitat. Once we collected the organisms, the samples were extracted in a solvent. We use ethanol. And then through, I think of it like a sponge daiquiri or a sponge milkshake, but with alcohol. And then through a process that we called bioassay guided isolation, the bioactive components of that extract were identified and then ideally patented and licensed for discovery. So think of it like this: the sponge or the gorgonian or whatever has hundreds of different chemicals in that extract, kind of like a cup of coffee. So what we're looking to find is the caffeine. What's the active component of that extract? Next slide, please. There are a lot of bottlenecks to this approach, not least of which is access to genetic resources of the country within which the samples are collected. It's often very difficult to get permits to do collection, to remove a country's genetic resources. Of primary concern are conservation, making sure there's no damage to the environment, and then how the host country would benefit should a drug be discovered and developed. That's the topic of a whole other presentation, but I'd be happy to answer questions if I can after our presentations today. Typically, the chemicals are present in very trace amounts, so ensuring that there's an adequate supply for development of the drug is probably the second largest bottleneck. Ideally, a drug company who would be developing this would like to have total control over its synthesis, so chemically synthesize that compound. But the molecules are often very complex, and their synthesis can be quite expensive. Still, if it's really a potent drug for a disease for which there's no treatment, they'll spend the money. The drug companies will spend the money to synthesize the compounds. Other options for production of the molecules can be fermentation, if a microbe, for example, is involved in the biosynthesis of it, aquaculture, or even cell culture. Wild harvest is generally not an option for obvious reasons, the ecological damage that could occur, and also how expensive it is to get out to sea. Our research group is working on cell culture production of sponge-derived chemicals. Again, the topic of another presentation, but I have a few slides on our research if anyone is interested in discussing after the presentation. Next slide, please. I'd like to shift focus now to introduce you to a European Union Horizon 2020 funded project called Project Sponges that involved more than 20 collaborating institutions from Europe, Canada, and the US. The goal of this 4-year project was to explore sponge grounds in the North Atlantic, document their biodiversity, and evaluate the ecosystem services they provide in an effort to conserve these vast areas of biodiversity. On this map, all those white dots represent areas where large masses of sponges occur. One aspect of the project was to develop alternatives for sustainable discovery and supply of sponge-derived bioproducts. And the organizations involved in this aspect, the marine biotech aspect of the research, are indicated by the logos on this slide. Next slide, please. Metabolic diversity of the key sponge ground species was assessed by basically fingerprinting, metabolically fingerprinting to identify new bioproducts with industrial potential. And so an integrated omics approach, so transcriptomics, in general, genomics, was used to search for pharmaceutically relevant genes and gene clusters. Sponges are also host to a diverse array of microbes, a microscopic image of which is at the center left. Dr. Manning is going to be talking about microbes in his presentation. In addition to the bioactive compounds, deep sea sponges also produce very intricate skeletons. It's very cool. The image on the lower left is a scanning electron micrograph of sponge spicules, which are microscopic siliceous skeletal elements. And these are from one of the sponges that occurs in these massive sponge grounds. These skeletons are actually serving as inspiration for research on bone tissue engineering. And in addition to this, what's even more interesting is that mimics of sponge-derived silica are potential biomaterials for bone tissue regeneration. So the cartoon on the lower right, which you may not be able to see very well, actually represents cells that are involved in bone tissue formation. In cultures of these cells, bioactive sponge, like similar to sponge-derived silica, attracts and stimulates these stem cells to differentiate into cells that synthesize bone. Next slide, please. So the major biotechnology outputs from the project underpin a more sustainable, non-extractive approach to discovery and development. So we've identified a number of bioactive compounds, but looking at it from not from collections, but mining the molecular diversity. And we've created open access databases of the enzymes and the gene clusters so that others can take advantage and mine this database. We're evaluating the potential of sponge-inspired silica-based materials for bone regeneration, and we're looking into sponge-inspired architectures as tissue engineering scaffolds. And finally, our group developed the first marine sponge cell line which we're using for in vitro production of bioactive materials. Thanks very much. I'll turn it over to Dr. Manning now.
Shirley, thank you. And please stay around because we'll have some questions afterwards. And I've got some questions for you myself. Tom Manning, professor at Valdosta State University, is doing some amazing work. And what really strikes me is that they're already in preclinical trials to use these materials for cancer, TB, and COVID. So it's really, really important work that's going on. And Tom, tell us about what you're doing. Tom, you're muted.
Hi, my name's Tom Manning, and I'm at Valdosta State University. Could I get the first slide, please? Okay, so our work focuses on synthesizing drugs, but we let bacteria in the ocean do it. The drug I'm going to talk about the most today is called bryostatin, and bryostatin is in some ways considered the flagship for marine natural products or drugs from the sea. It has efficacy against multiple sclerosis, against Alzheimer's. It has FDA orphan drug approval for Fragile X syndrome. It also has anti-cancer and antiviral properties. Its problem is that it's very, very difficult to synthesize. And so right now its cost is on the order of $20 million per gram. And so what we want to do is to come up with an economical synthesis. So right here, this is becoming a diversity issue. In the ocean, if you take a liter of seawater, it contains up to 10 billion bacteria and up to 100 billion viruses. And there's a dynamic there between the viruses and the bacteria, where the viruses want to inject their genetic material into the bacteria, and the bacteria fights them off with a chemical defense system. So this is our method that we're optimizing, trying to optimize for making drugs in the ocean. And you'll see that we're not just, we do things in a very small scale, right? And we still get decent yields. And it's much different than as a chemist I would do in a lab. Many of the things done in a lab can involve, it can take months to do a total synthesis. It can involve very expensive equipment. You need to hire fairly expensive, highly trained, very skilled organic chemists. And so we're trying to simplify it by what we do, what we call pharmaceutical aquaculture, or farming the sea. Next slide, please. So I should point out that a problem here is, Shirley just talked about that, is that the ocean is a reservoir for new drugs. The question is, how are you going to get them out and how are you going to learn about them? So the first thing I want to do is to give you a visual example of what we primarily deal with, and I call it a biofilm. It's mostly bacteria, but there can be other microbes in there. On the left side, you see a piece of pine, fresh cut pine, not treated, that was in the ocean for 2 years. And what you notice on it is that absolutely nothing grew. Okay, so pine has a built-in chemical defense system, just naturally occurring, that for whatever reason keeps things like barnacles, different types of fouling organisms, biofilms from growing on it. On the right, and this is stuff that we do with oysters, on the right you can see pine would also, but we've treated this both physically and chemically, and we were able to selectively grow oysters on it. So the key point here is what we're after is materials that will attract biofilms. And by adjusting the chemistry of the surface, we can adjust what type of biofilm, what type of bacteria show up. And again, in the ocean, nobody knows the exact answer, but billions or trillions of species of bacteria, and unknown number of species of viruses. And so we just use biofilm, and we don't try to figure out what's in the biofilm because we can't, but that's what we're after. Next slide, please. Okay, so let me give you kind of our approach. And this is a philosophy that seems obvious when I say it, but it's important. When we come up with our nutrient base, what we want is all the ingredients for life of what we're after. Okay, and so what we don't want is a standard agar that might be used in a lab, because many marine bacteria do not or cannot be grown in a lab. They can only be in the ocean, where there's this complex chemistry, there's this complex dynamic with other microbes, and as soon as you take that bucket of water or whatever you're bringing back to the lab, things change. And just a simple example, right? You have an orange, eggs, and potatoes, right? They all three of them grow in different, hosts, right? The potatoes on the ground, the orange is hanging from the tree, the eggs inside a chicken. But if you had a bacterial infection in an egg, it wouldn't impact the potato or the orange, right? And the nutrients that, say, an orange needs is not the same as what a potato or egg needs. So what we do is we try to adjust our nutrient base and our location based on a type of bacteria that we want to grow, in the same way that you would get any one of those three. So on the right is one of our materials, and the basic idea here is that has specific nutrients in it that went into the ocean. In this case, it will stay there for weeks or months. It will get partially eaten and devoured by bacteria. It's in a container to keep out light. We want to minimize photosynthesis. And it also has the water that comes and goes is through very small holes, because we don't want predators like fish and crabs coming in and eating it. And then we'll take this out and we'll extract it. And so this is very flexible. We can change the nutrient base. We have some inorganics in there, surfaces that we can change. And this is what we're using. We're focused on bryostatin right now with this method. Next slide, please. Okay, so let me give you two examples. So there are two kind of classic marine natural products that pop up quite a bit. One is called ET743. It has a trade name, Yondelis and Tribectin. And it was originally extracted from a sea squirt. And it took on the order of 95,000 pounds of the sea squirt to get something on the order of 2 ounces of the drug. And this is a tiny creature, right? So it's not this, massive glob under the ocean. So that was an incredible effort to do that. But it does have efficacy. The second one, the bottom one, is bryostatin, which comes from bugula. The first large-scale harvest for bugula was 14 tons of this small bryozoan, and they got half an ounce of the drug. So as you can see, both of those ways are not sustainable. So in the exact same ecosystem, we set out two different nutrients, and I can't go through all the details, but some of the highlights of the first one, it was an acidic nutrient that had a high sugar content and a high lipid content with some other things. And we were able to get ET743 and a couple of other ecdicidian molecules that have been published. In the bottom one, we had a different nutrient base. It was a neutral pH. It was high in starch. It was also high in iron and calcium, limestone particularly. And that replicated the outer coating of the bryozoan that we use. And we got several bryostatins. So the point is as we adjust this nutrient, we can adjust what microbes grow there, and we can get different molecules out. Next slide, please. Okay, so this is a lot of the work we do, and I'm not going to talk about mass spec or NMR too much, but it has to come up in at least a visual way. So on the bottom is LCMS from our lab, and it's for bryostatin 1. Bryostatin 1 is the one that has gone through for FDA approval. It's used in clinical trials. It's the one that costs roughly $20 million per gram. So it's extraordinarily expensive. But you see one molecule there. On the top, you see one of our extracts, right, where we did the extraction a little bit different. And what you see is multiple molecules that correspond to different versions of bryostatin, right? So bryostatin structure can change slightly. And so what we're starting to think or see is that and we can do calculations for the ones on the top that measure biological activity. How does it impact a kinase? How does it impact an enzyme? And each of those different bryostatins have different activities. So in the same way that like a jellyfish sting, if you look at the chemical composition of a jellyfish sting, it has lots of different molecules and proteins in there. It's not a single molecule. We're starting to investigate if, when we look at bryostatin, should we be testing for one molecule, or should we be looking at the combination to see if it works better? Right? And there's some precedent in nature for that. It's also a bigger thing now, like for example, in oncology with cancer drugs, they're starting to learn that you can use 2 or 3 molecules that attack the cancer in different ways, works better than a single molecule. So right now, like in our lab, we have things set aside where we're pushing to have the mixture tested, for cancer activities, and then move on to other things. Next slide, please. Okay, so let me leave you with a thought experiment. right, and how something like this might work. So when we do our work, we're in fairly shallow water along the coastline. We pick our locations. There's a specific, actually a number of specific reasons why we end up in one place, and we're doing this on a fairly small scale. But when you think of a farm for drugs, we're not talking about, 1,000 acres. We're talking about an area that might be 20 feet by 20 feet or 30 feet by 30 feet, fairly small, right? But close to the surface for bryostatin or ET743. But just from a discovery point of view, let me give you another perspective. So let's say we're over the Marianas Trench and we have the ability to drop a steel cable to the bottom, and to this steel cable we put metal boxes, and they're perforated, and each metal box has different nutrients in it, right? And maybe they're spaced out, say, every 100 feet or every 200 feet. We let it sit there for a month or two. We come back, we pull it up, and then we start doing our exploratory work. We look for, new drugs, but there could also be new herbicides and pesticides in there. There could be new types of proteins that we haven't seen before, right? There's glues. A lot of marine critters attach themselves to surfaces with glues that work extraordinarily well. There might be unique metal ligand complexes that can serve as a catalyst, or can be a method of converting chemistry into energy. So the point is, there's a whole, with all those bacteria, the viruses, their interactions, all the other microbes that are out there, there's a whole frontier that can be explored. With our method, what we're trying to do is, with bugula and with the C-squared, Ectocidium turbinata, I mentioned how many thousands of pounds of material have to be garnered in order to try to get a very, very small amount of drug. That's not sustainable. It's not green. Our approach is meant to be a green technology where the nutrients we put into the ocean are already there. They're just concentrated. And when we remove something, there's no footprint. And we're not harvesting, right? We're not going out harvesting bugula. We're growing stuff on our material. And so that's kind of the future look ahead. I will say I'm a chemist. And so if I gave a chemistry talk, there'd be way too many NMR spectra and mass spec. So we kept it kind of from a philosophical point. And I would be happy to answer any questions when the time comes. Thank you very much.
Thank you, Tom. So this is fascinating, and I'm really fascinated by how much material it takes to get such a small amount of work. So your last slide was really interesting to me. Let me ask this. We have a question from Kathy Sakis, Dr. Papani. Is there any interest on sponge farming?
And sponge what? Oh, sponge farming. Yeah. Actually, there's been a fair amount of effort put into sponge farming over the last couple of decades, actually even going back maybe probably as early as the mid 20th century as well. So there's some, for both in the sea and also sponge aquaculture, so land-based systems as well. We're in fact working on a project where we're trying to develop little small sponges from our sponge cell cultures. But yes, there is interest in that, and it's been successfully done in a few places, both for production of for harvesting for compounds, and also for bath sponges.
Okay, and so that's an interesting question. So I hadn't thought of that, but the bath sponges do produce these other chemicals that we need in them.
Yeah, sorry, you're kind of breaking up, so I didn't hear the whole thing. But yes, there was a group that actually farmed a sponge in New Zealand, from which they extracted a fair amount of material that's gone into a drug that's clinically available right now.
That's great. For audience members, if you can hear me, and I'm sorry for my audio, if you have any questions, put them in the questions section. I'd be happy to ask them. Shirley, do you have any questions for Tom from your side?
Oh yeah, I have lots actually. I found this really fascinating because it, I think we've both been involved in this for a number of years, and so I mean, for sure, bryostatin, the bugula was initially they got that from wild harvest, as you mentioned, and the same thing with the Echinacea, the Yondula. So I think, your approach is really super interesting. It kind of reminded me of some of the work way back in, I think, again, the mid 20th century that Ruth Turner did, where she actually would put, pieces of wood down on the ocean bottom, and she was really specifically interested in these wood boring, bivalves, I believe. But it's it's a very, very interesting approach. And also, I find it also interesting. I mean, I'm glad you brought up the point that it's not only drugs from the sea that these marine organisms can provide, but enzymes, fine chemicals, pigments, antiphthalates, even nutritional supplements. Do you want to talk a little bit more about that?
I could mention it because that's one of the, because people go after pharmaceuticals because there's the greatest need. I mean, everyone sees immediately, oh, if you have an anti-cancer drug versus a pigment, which one are you going to go after? But my own perspective, which is, speculatory, is that there's so much to be learned. I mean, because a lot of times when people go down, I'll give an example, and the numbers are not going to be right because no one can give you exact numbers. But when you scoop up a handful of sediment, right, there can be upwards of 10,000 species of bacteria in there, of which maybe 5 or 10 are dominant and the other ones are inactive. But if you switch conditions a little bit, then something else can happen. So the point is, when you look at that diversity and you look at most of those things defend themselves with a chemical system. And just so for people that might not know when I say a chemical system, a great example of that is the tobacco plant, right? And it defends itself with nicotine, right? So a lot of things in nature will not go out and eat tobacco because it has nicotine. We smoke it, put it in our bodies, but everybody else in the world recognizes it as a poison, right? So that's the same thing that bacteria does, right? A lot of our antibiotics have come from bacteria that you can grow in a lab or fungus you can grow in a lab. But the exploratory part on the planet, on the terrestrial part I don't want to say it's over, but a lot of people are looking for new sources. And the other things that can happen, I mean, there can be proteins that can be used in a host of different ways that have yet to be discovered. And there's probably, an untold number of them. And things that live in, under deep pressures have a different chemistry than they do at the surface. If you go down further and further and further. So with those, it's just kind of so wide open that you could come up with 100 ideas, and you might be lucky if you could test one of them. So
there's a question that just came up on the cost of the size of the market for sea drugs. Shirley, 2.3 million was it? Billion?
Billion, yeah. Well, I mean, you can think about Yondelis, derived from just the two examples that Dr. Manning provided. So the bryostatin and the ET743, both of those, I mean, are fairly sizable in terms of the revenues that come in. So you can understand why host countries, for example, are really interested in what kind of benefit they would get from any kind of genetic resources from which drug leads that are eventually developed, happens. What happens? How are they compensated for sharing those genetic resources?
Okay, there's a question. Someone says, I'm interested in permits to take samples back to the U.S. Does that make sense to you?
It does. First of all, probably going to be hard to get even for people to get back into the U.S. for a while, but. Right. Yeah, I mean, there's a lot of permitting. The initial permit is really a request to the host country to be able to access that material, to be able to collect it, to be able to export it, how you're going to involve the scientists from the host country if they're interested as well. And then coming back into the United States, depending on what it is that you're bringing back in, you'll need to get, if it's something that's on the endangered species list, that's a whole other layer of permitting. And usually with fish, you have to detail this with Fish and Wildlife. I have to tell you, we haven't done a lot of collections outside of the United States for a long time because of how difficult it is to get permits.
Ivan Francis asked a question. He says, I read a report on the Aquarius reef base is able to show that 10 days of research diving from the habitat provides about 6 months of research based on diving from the surface. Is there an application of using underwater habitats like that to discover drugs from the sea?
Well, I have some personal experience there. I spent 9 days in the Aquarius, and it was probably one of the most remarkable professional experiences I ever had. And yes, I think that underwater habitats have an application if it's something where you want to conduct that research, and like, for example, the work that Tom's doing, I think would be particularly amenable to that, because it would give him, give that type of research, people doing that, access on a fairly regular basis to go out and do like sampling, where if you had to go and dive, it might be more prohibitive. You're only allowed to, you can, whereas if you're in a habitat and you're saturated, you can stay, out a lot longer, you can go out more frequently. So there are specific applications, and you can set up experiments that you can monitor on a, fairly continuous basis as well.
Ian Gardner mentions that he's involved with the Ocean Genome Legacy. Do you know that lending library with 28,000 samples and collecting data? Can you tell us a little bit about that? That's interesting.
Yeah, I mean, that's interesting because, again, looking at doing like molecular fingerprinting, and you can start mining these genomes for specific gene clusters that are already known to be pharmaceutically relevant, as well as kind of doing some exploration. Tom, you might be able to add more to that as well.
I'm not a geneticist, by training, but I would think it's critical. One of the large-scale efforts that went into bryostatin was out at Scripps in San Diego, and they tried to move the genes out of the bacteria that was in the ocean and into, I think, E. coli, something that would grow in a lab, but they were unsuccessful. But that genetic information, I mean, if you can start moving genes around because it's very, very hard to grow marine bacteria in a lab. But if you can get some genetic material out, move it somewhere else, and then, have a broth, basically, that's one way of, mass producing the pharmaceuticals. There's just been a lot of, and I can't tell you on the genetic level what the problems were, but they had a lot of, it hasn't been that effective, but it's possible. So I think the more work in that area is more likely to lead to, large scale production of drugs or other things.
Ian points out that the OGL was started by the New England biolabs, and he says that they synthesize genomes and may be helpful in this process. So we have another question from Kathy Sakas. She said, marine life and especially sponges have natural chemical defenses, as you mentioned. So where are these applications currently being used in agribusiness?
I don't know of any right now. I know many years ago we were working with a company and they did some screening as kind of natural pesticides, but nothing ever came out of that as far as I can tell. But that's another area that I think is ripe for development. And I wanted to just point out one other thing too, because the timeline, and one of the challenges I showed on one of my slides was that the timeline from discovery, basically from, seafloor to sickbed, from collection to the clinic, tends to be very long because there are a lot of steps in that, the FDA approval process. And I always kind of counsel people, think about a diversified portfolio. So you don't want to put all your eggs in the drug development business. There may be other approaches, for example, antiphallants don't take long to get approved, some topicals, cosmetics, things like that. So that's why I think we're probably underestimating the value of marine natural products in industry.
Well, and your last slide sort of dealt with that, looking for not just the biostatin, but the other applications that could come out of the sample.
Yeah, if you think about when they mass produce a drug, Taxol is a good example. It's a cancer drug. It's the best-selling cancer drug over the last 30 years. That they estimated they would need 100 pounds a year for the world supply. When you deal with agriculture, 100 pounds might be one farm, right? One decent sized farm. And so you have to get to a point where you can mass produce tons of the material, I think, to go in that direction and to attract people. And so that's where an economical synthetic process lies, where its value lies. It's just to be able to produce lots of it very, very cheaply. If you look at most herbicides and pesticides, they're fairly small molecules that are easy to synthesize. And so that's the same type of thing, that's just more looking at what's coming out of the ocean that's economical. With medicine, you have there's more freedom with it being fairly expensive because, it's more important in terms of what a small dose can do.
Can you talk a little bit about what you're doing with the COVID research?
I could tell you that in a nutshell, we jumped on that early because we were doing work with inhalation with some of our tuberculosis compounds. So if the viral load in someone's lungs is where you're sick, right? So if you can knock out the viral load in the lungs, then you have something. And so what we did was we developed a compound, or I should say a mixture. We published a couple small papers on it. A doctor in Mississippi, well, first a doctor in Iran tested it. It worked well. We went to a congressman that's a pharmacist, and when we said Iran, he said, that's it. We're not going to talk about that anymore. So then we got lucky, and a doctor in Mississippi did it, and it worked very well. He had 83 patients that were, hospitalized for COVID. We published a small blurb on that, and then we ended up filing an application with the FDA for what they call the EUA, emergency use application. It was myself and the doctor. There was no big pharma with us. And it got turned down predictably, simply because, part of my interpretation, which is subjective, is we just had no resources behind us to do it. So it went well. The results were turned over to FDA. It worked well, but it attacked it. It was an inhalation. A key part of it was we had to make our particles very, very small, nanoparticles to penetrate deep in your lungs. And so that was a big part of it, because normal inhalers or whatnot only get to your throat or your upper lungs. And so we were making particles that were 50 to 100 nanometers in size to get down there. So the goal of that was to denature the spike protein that's on the outside of the coronavirus, and that's what it ended up doing. So,
wow, interesting. So you mentioned the in vitro, growing the cells in vitro, Shirley. How scalable is that?
Actually, and that's what we're working on now. We've been able to develop, well, first of all, just getting the cells to divide, it was probably took me about 30 years, but we got the cells to divide finally. And so we can get them to grow really rapidly in culture, but being able to scale up enough so that you get adequate biomass is the challenge that we're working on right now. But what we're doing is we're encapsulating the cells in small droplets and being able to put them in bioreactors, and the cells grow well in these small gel micro droplets. And that's actually what we're working on right now. That's the focus of our research. So hopefully we'll be able to scale up in vitro. And as I mentioned, we're also working on aquaculture, production of biomass through aquaculture of these sponges that are grown from cells, rather than going out and harvesting perfectly healthy sponges, cutting them up into fragments, and then trying to grow 10 sponges from one that we just killed. We're trying to do it kind of an order of magnitude more using just the cells from a small portion of a sponge.
Well, that's wonderful. And I feel like we're on a real leading edge conversation here, and it's so exciting to have you as our guest. And I'm going to turn it over to Millie.
Millie, we can't hear you.
Yeah, unmuted now, right? I have one last question, and either or both of you can answer it. Will the attention to this attract unscrupulous people or countries to start harvesting the natural resources in a way that your work absolutely is unintended for? And you've both mentioned this, the sustainability of doing this kind of natural harvest. What are your concerns about that, and how is that protected, or is it not protected?
I can start, for sure. I think that probably the weakest, I shouldn't say this, but probably the weakest level of protections is probably in the United States, because it's fairly easy for people to, there are regulations that are established around each state in terms of getting permits to do collections, but I don't think it's policed as well as it needs to be, just because of lack of resources. In most countries, you need to get permits to even do anything, do any kinds of collections or exporting. So I think that in itself is one deterrent. Also, the cost of getting out is quite expensive as well. Now, one of the questions I'm asked a lot is, what about out in the open sea, going into the deep ocean, beyond national jurisdictions? And again, that could become an issue in the future, but right now I think that's also cost prohibitive. It's expensive to get out to the deep sea.
Okay, well, that's comforting. Tom, you have anything to add to that before we close out?
Yeah, I point out when they first started doing harvesting of bugula and arantina for browstatin work, they used shrimp boats. They rented them, and they would scrape the bottom, and the bycatch rate of everything else that was there was greater than 99%. And bugula is this little tiny thing the size of your hand, you know, kind of like, it looks like seaweed almost. And so to get 14 tons of that, you can imagine how much ecosystems, you know, how much acreage was just absolutely destroyed to get that little, that tonnage of bugula. I know that they've also done it off the coast of Mexico and off the coast of Vietnam. And so that's just not a long time. I mean, because once you destroy the habitat, it doesn't come back. And so might be able to get the drug for a little while, but then your supply is going to run out and, you know, game over. So.
Right, and that actually reminds me too that there are other threats that, you know, not that Tom brings up, that it's not necessarily related to these organisms that we're interested in, but as bycatch, and also deep sea mining and drilling, things like that. We've demonstrated through the Project Sponges that these, like drilling muds, things like that, really affect these organisms. They affect the biodiversity, and once gone, they're gone for, you know, for decades or even centuries.
Great. I share Dane's comment. This does seem very leading edge. A lot of us in this community talk about the long timelines to get things to market. And more typically, we're talking about some hardware, maybe for new energy. This takes that conversation to a whole different level. I mean, the timelines. You said, Shirley, how many years it took you just to get some cells to reproduce. Oh my goodness. Anyway, thank you both. This is great. It's a great way to close out our year. I want to again thank the Marine Technology Society, and I want to recognize Dane. Dane is the founding partner of Reservoir. He does all this for us pro bono because his love of innovation and sort of deep thinking. When he's not doing this, which is most of his time, he does a lot of work with leaders and helping them develop to be the best they can be. So if you're interested in that, look at Reservoir. Next slide. Watch for what we're going to do in 2022. We haven't determined that yet. Maybe we're going to take a month or two break from this, and then we'll come back with something new and fabulous. You see Ocean Exchange and MTS websites here. I encourage you to explore both of them and find out more about both of us. Next slide. Send us your feedback. We've gotten feedback over, we started a webcast in 2020, and then we teamed up with MTS. If there's a topic that's about marine technology innovation that you'd like to hear about, let us know. Next slide. And then thanks to our panelists, again, thanks to Dane. We have 3 terrific people behind the scenes. Alice is with Ocean Exchange, and Monica and Christina are with the MTS. We curate these. It's being recorded. If there's something that you have a colleague, someone in the business that you think should review these webcasts, please share the links with them. Thank you very much. Happy holidays to those where December is an important month for you. Happy New Year. We look forward to seeing all of you in 2022. Thank you.
Thank you.